our next guest is actually, I'm going to get to face a fear that I've had since I was like, knee-high to a grasshopper. And I am really afraid of frogs. Okay. However, our next guest, Tyrone Hayes, is going to tell us why frogs and all those other amphibians are our friends and why, and going to unlock some mysteries about them and how it relates to the rest of the world. Tyrone?
So, what I'm going to do first, before I tell you anything, before I ever give a talk, is I always give my acknowledgments or my thank yous first. The reasons, if you will, for how I came to be here anyway, in the first place. And my first acknowledgement is always to my wife, Catherine Kim, and my son and my daughter, Tyler and Cassina, and my parents. Because regardless of how cool my job is, I would be nothing and nobody without these people in my life. So to my family. I couldn't do my cool job without money. So these are the organizations and that have funded me. And I also couldn't do my cool job without a lot of cool students who really worked really hard. And so this is a list of the students that I've worked with over the last 10 years. And everybody in blue is an undergraduate college student. And this is what I call my new crew. There we go, my new crew. And one of the other things that's very important for me and in my job is the celebration of diversity and the diversity that I get to enjoy in my laboratory, as is illustrated here. And finally, just one more to my family, to my grandmother in particular, who passed away 2 Christmases ago. It was her dream in life to make the world a better place through education, and I'm thankful that she passed that dream on to me. And I have to say, with all of my fancy education at Harvard and Berkeley, et cetera, the most important things I learned in life, I learned from the people that I grew up with in my house, such as my grandmother and my parents. So what I'm going to try to do, I'm told I have 12 minutes to basically try to summarize 42 years of life. And that comes to, I think, about 3.5 years a minute. And it all starts with this book. This is the very first book my mother ever gave me. At least that's what she tells me. I don't remember the book, but my mama don't lie. She says this was my first book. This was my first book. What I remember, though, is being in love with amphibians, being in love with biology, which literally means the study of life, being in love with life for as long as I can remember. And this question, what is a frog, is really one that I've been trying to answer for as long as I can remember. Now, if we can switch to the video clip, I want you to pay attention. I want to show you the thing that fascinated me about frogs. And just watch. These are eggs, and this is a time lapse. And if I have to do 3.5 years a minute, this is 7 days worth of development in 30 seconds. That's what fascinated me. And I'll walk you through, in a less fancy way, I'll walk you through in some of the photographs that I've taken in my laboratory. What you were looking at are frog eggs. They start off, they're going to end up like this, but they start off as single cells, the size of a pinhead. And what you can see in this photograph, this is obviously blown up, you can see the corners here. This cell is already divided into 4 cells, and it's starting to divide even more. In a few more hours, it'll look like this. So each one of those sections is a cell derived from the fusion of 2 gametes, from a sperm and an egg. Here now is more. In a few more hours, actually in about a day, each one of those little dimples is now a cell. This big ball of cells will undergo what's called gastrulation. That'll create the gut and the skin, and then it'll go through neurulation. As you can see here, it now is developing its spinal cord and its backbone. And in a few other hours, few days actually, it'll develop into a tadpole, a living, swimming, breathing, functioning organism. Now here's what excited me. It's not all that different from how you and I developed. We undergo gastrulation and neurulation, development of our nerve cord, just like these frogs do. We now know that we know so much about genes that the same genes are involved. You formed your spinal cord the same way, that same genes, using the same genes as this animal. The difference is, and the difference for me when I was a child, was that you could see it. So my little brother developed inside of my mom before there was ultrasound. So I didn't get to experience the miracle in the same way. If you're a chicken, you develop inside of an egg, you can't see it. But if you're a frog, there's no eggshell, it's not inside of mom. You can witness, you can see, even without a microscope, many of the events that I just showed you. Here's the other thing that made the frogs more interesting. They had this extra developmental step that we don't. They metamorphose. And what's fascinating about that is they go from a completely living, swimming, breathing, functional organism, and then they tear that apart, and they build a whole new organism. And I mean literally. So literally inside of that one egg, you have all of the genes to make a tadpole, and then you have all of the genes to make a completely different organism. And it does more than what might be obvious to the eye, right? The tail goes away and it grows legs, and the gills grow away and it grows lungs. But every single gene in that animal is being turned off, and a new set's being turned on. The hemoglobin's different. All of what are called the cell surface proteins are different, such that if I clone this frog and then tried to do a tissue graft from the frog back to the tadpole's immune system would reject it. It wouldn't even recognize it as the same organism. Now, it turns out that all of those genes being turned on and off are regulated by chemicals called hormones that these frogs make in their bodies and that we all make in our bodies. And it turns out that my interest as a little boy in these animals, because they were accessible, is really the same interest now that I have as an adult. Because it turns out, because they have no eggshell, because they have no placenta, because they have no membranes, any chemicals in the water that might interfere with those hormones, what we call endocrine disruptors, have direct access to this organism. And so it makes a good model for the kinds of things that I want to study. Now, the other thing that I was fascinated by as a child was going to Africa. I would watch National Geographic and David Attenborough, and I never got on a plane until I went to college, so I didn't get that opportunity. But as an adult, as a little boy who grew up and was still interested in science, I got to go to Africa. And while in Africa in graduate school, I discovered this little group of reed frogs. They're Hyperoleus is the genus, if you're interested. And it turns out that everyone you're looking at here is the same species. And so I get fascinated in not just how they develop and how they metamorphose, but what are the genes and what are the hormones that determine whether you're going to develop yellow with big blue spots or whether you're going to develop little polka dots. What's more is I discovered in a place called Sokoke in Kenya that in some species, the males and the females look like completely different species. And it turns out that they all start out, so these are the metamorphosing individuals photographed once a day for 6 days. It turns out that they all start out green, and if you apply testosterone to the water, there's no effect, but it turns out that estrogen will make these animals change color and become the female color. The same estrogen that regulates reproduction in all of the women in this room. Exact same hormone. So one day, talking about paths, one day I was giving this talk, and my wife was in the audience, and she said, well, why don't you patent that? I said, what did you mean? Well, you can't patent a frog. And she called her brother up, and he says, yes, you can patent a frog, heretofore to be referred to as the so he's a lawyer, so he said all kinds of fancy stuff. And we patented it. We called it the Hyperoleus argus, that's the species, endocrine screen, endocrine means hormones, or the Hayes test. And you might wonder, why would anybody patent a frog? Well, it turns out that all of the estrogens that make this frog change color are exactly the same chemicals that promote breast cancer. So we had a little test where we could dip a frog in a solution, and if it changed color, we had a potential cancer-causing agent in that water. And then a funny thing happened on the way to the shop, as they say. I not only went now from a little boy just interested in frogs and trying to do quirky experiments to understand frogs, but I sort of became an entrepreneur. In that process, a big chemical company asked me to study this chemical called atrazine, which I had never heard of. It's an S-chlorotriazine. I hated chemistry as well, and now I have to look at molecules all day. It's an herbicide, or it's a weed killer. It's been used for 48 years, so it's been around longer than me. And we use 80 million pounds a year. If you can imagine, 80 million pounds per year in the United States. It's used in more than 80 countries, so it's a global issue, but it's now outlawed in all of Europe. And I put this up here, the words, because the company responds interestingly when they see the words. Like they threaten to sue me for putting these words here because they say, well, it hasn't been outlawed in Europe. It's been denied regulatory approval. So whatever the difference is, they're not thinking about the real science. and the implications. At any rate, I want to tell you briefly how a little boy does the kind of CSI stuff in the laboratory and still enjoys himself in the things that he likes to do. We look at laboratory models. So for example, we have frogs that we raise in the lab called African clawed frogs. And we've shown that when they're exposed to this chemical, they produce a number of reproductive abnormalities. So for example, single-sex polygonadism, all it means is that some males develop multiple testicles. And so in fact, this picture ended up on Jay Leno. He made some comment about a frog has 6 testes. And he said something like, what's the problem? What guy wouldn't want a few extras? And I know that the audience is a little bit younger, but trust me, 2 will get you in enough trouble. We found frogs, though, that were male on one side and female on the other. We found frogs that were male in the front and female in the back. Try to figure out which restroom you use in that situation. And we found frogs that were all just mixed up, multiple testes and ovaries. But in any case, none of it's normal. And by that I mean frogs did not normally develop this way. This development is because of chemicals that we're using in agriculture to grow food that are ending up in the water where these animals are trying to survive. We now have a new publication coming out where kids close your eyes. That one frog is not helping that one because he hurt his leg. In fact, these are 2 males. These are 2 brothers. The one that's performing as the female is a genetic male that actually lays eggs. So again, I'm not making any kind of judgment. I voted for Prop 8. Most of you don't know what I'm talking about. But the point is, these chemicals that we're putting in the water are causing a hormone imbalance. And whereas the genes for that female tells it that it's a male, these chemicals are making it become a female. And that should be a concern. Well, we took our laboratory model and other work that we published. We did what I call comparative studies. We looked at a North American species. And what we found in North American leopard frogs was that these males develop, so that is the testis you're looking at, but all the stuff you see at the back, what I call the junk in the trunk, these are all eggs that are bursting through this male's testicles. Now, I started working with the Environmental Protection Agency over these issues, and I sent them this photo, and I said, look, this chemical is making males grow eggs in their testes, and they wrote back and they said, well, that's an interesting finding, but we don't know if it's an adverse effect or not. When my wife was having our son, she had this, what I call G.I. Joe Kung Fu grip on my hand, and she later told me that that was the most painful experience she ever had, and so I can't deny that, but I would have to guess that a dozen chicken eggs in my testicle would be a little bit uncomfortable, and that the Environmental Protection Agency would be concerned, but that's not what's happened. So nevertheless, we published it, and then we got to do these field studies. So here's how I satisfy myself. I can answer these puzzles, I can answer these interesting questions, and then I can go out in the field, out into the wild, and ask, do I see these kinds of abnormalities in the real world? We get to go to great places like this. This is a pond, a lake that we had to hike a week to get to across 2 mountains in the Sierras. Here's what I want you to understand. Even in this remote area, we can still find pesticides that get picked up by weather patterns and drop down in these remote areas. We can still find frogs infected with diseases that originated in Africa. That's the kind of global impact that we're having. A place that you have to walk a week to get to, you still see the signs of our impact on the environment. They're global. The other things that we get to do, because we start in the lab, look for the problems in the field, and then go to the field, is we get to do things like understand better how to design our laboratory experiments, what I call field simulations. So for example, if you go to a site like this, where the runoff from that corn, which is where atrazine is used, is exposing those frogs, what we find is farmers use many, many more chemicals on any given field. In fact, that's just on that one field. And so what that allows us to do is to do more realistic experiments by testing multiple mixtures of chemicals. And so you can see why there's so many undergraduates involved. That's a lot of frogs you got to take care of. And what we find are things like not only do you see sexual abnormalities, but these pesticides increase their stress hormones so that they get diseases like on the right here. You get diseases like flavobacterial meningitis because their immune system can't function and fight off the diseases. So you have an interaction of effects in the environment that are contributing to global amphibian declines. What we really want to do now is try to address how do you in a complex environment like this, and this is over in Nebraska, how do you in this complex environment address how all of these factors, depleting ozone, increasing global climate change, habitat loss, pesticides, disease, how do you really understand what the real impacts on amphibians are? We do that by, and I call this our lab, field, lab model, because we start in the lab, we go to the field, we design some better lab experiments, and then we literally bring the field, the wild, back into the laboratory. So, and this is the kind of stuff little boys dream of. So for example, here are two students and I, we packed up tens of thousands of gallons of water, we rented a giant freezer truck, and we drove it across the country from Wyoming back to California. And in other years, see, it's the same site, just a different year, when there's no contamination, we do the same things. So we can actually literally test the water inside the laboratory under controlled conditions. And that's how we complete what I call the lab, field, lab model. The other things we do is we run the model backwards. We actually bring the lab into the field. And here's what I mean by that. So in California, where the Salinas River runs this way, it becomes more and more contaminated because as you look, there's a huge amount of agriculture in the northern part of the river. And that huge amount of agriculture is because 50% of the US's agricultural products come out of California. And in fact, greater than 90% of the US's lettuce comes from around that one river. And so what we're able to do, when I say bring the laboratory to the field, is we just let animals grow up in the contaminated water in the field rather than bringing them in the laboratory. And you get situations like this. That big tadpole is upstream before there's agriculture, and those two little tadpoles with immune problems are tadpoles that are growing up in water that runs off of your food, that's contaminated with pesticides and fertilizers that comes off of our food. Now, what has also been a surprise to me is that this little boy who's interested in how frogs work has now gotten involved in public health and economic and political issues. Because it turns out, there's an interesting relationship. This is Lake Nabugabo in Uganda. And it turns out, for example, that they don't have a fancy EPA, Environmental Protection Agency, like we do. But if I told the people in this village that the water running off of their crop, which goes into these containers, which is their sole source of drinking, cooking, and bathing water, if I told them that those frogs had eggs growing in their testicles and no immune system, the environmental health, public health connection would be clear. In many ways in our country, this is my village in Oakland. That's where my water comes from. It's not all that different. I don't have to take a yellow container and go up there and get it, but it's contaminated with pesticides at levels that are well below what the EPA regulates. So we're drinking many of the chemicals that cause these hormone imbalances, and many of these chemicals we now know are associated with things like increasing breast cancer mortalities. One of the bigger issues is it's not just us, the people who are drinking the water, but the agricultural workers who are exposed directly, who can have pesticide levels in their urine that are up to 20,000 times higher than we use to produce the effects that I've shown you. We're starting to get interested in something now called environmental justice and environmental racism. Because the people who grow our food, 90% of them are Mexican-American, and they have much higher rates of diseases like asthma, mortality from breast cancer, et cetera. They're also more likely exposed to these chemicals, for example, chlorpicrin, which was a nerve gas in World War II, compounds like atrazine. So who would have thought that a little boy interested in frogs would end up doing public health and at the same time still studying little creatures that he's just interested in out of curiosity. So I'm out of time, but I want to bust a quick rhyme. And my parting words for every boy and girl: if you love something, go to it. Or to bite a line, just do it. Who knows, you might end up saving the world.
Oh, please. Well, ma'am.
Have a seat. Ashe. Now, I have to say, I was really struck and actually deeply moved by, you brought your ancestors in, you brought your sense of history in, and that really, really resonated to me, as a Black woman, looking at a Black man, in a field where there honestly aren't that many, or there are plenty of us, but we aren't often, when they think of this kind of field, they don't think and understand of how diverse this is. So could you speak a little bit to how it has been for you, just even moving into this realm of work?
Boy, at the roots, my grandmother finished college and was a teacher. My mother didn't get to finish college. My father didn't get to finish high school. And one of the more touching things for me growing up in South Carolina, the young people here might not know it, but I was born in 1967, July 29th. I was actually pronounced dead July 28th because I was a breech baby, and at the time, blacks weren't allowed to go to the regular hospital. They had to go to the colored hospital. And I was so literally I have been not fighting since the day I was born, but since the day before I was born. And the interesting thing is I just lectured at University of South Carolina, where I grew up for the first time. And at the end of my lecture, my father was sort of in tears. And he said, no, it's not because of what you said. He said, because I remember that when I was a child being chased by the cops because it was illegal for blacks to be on that side of the street, where I was giving a lecture. And he said, now look at all of these people coming to see my son speak in a place where I was arrested. And that's had a big impact on me. And the places that my work have gone now, it's huge because, I don't know if you know, but blacks and Hispanics are more likely to die from breast cancer and prostate cancer. And there are no models, there are no cell lines, even to study why that might be. Right. And so we have the fastest growing segments of our population more likely to get and more likely to die from these diseases. And so being able to do that, when really I'm just a little boy studying frogs, but being able to apply that in a way and being able to challenge some of the big companies that won't acknowledge that, it's not, you just saw that really crooked path. It's not something that I ever thought about or that I would be involved in.
So you made that link. It's like I've been as an environmental justice advocate myself, and for those of you who don't know what that means, it really simply means that no community should have to bear the brunt of lots of environmental burdens and not share the benefits of some environmental benefits. And right now, race and or class, and perhaps sometimes definitely both, will tell you where you find the good things, parks and trees, and where you find the not so good things, like waste facilities or power plants, or working in areas where there's lots of agricultural runoff and exposure to chemicals that are going to have an adverse impact to your health. So you've clearly made that connection, and I want to thank you for giving me a deeper appreciation for frogs. I'll deal with that little issue on another time. But the connection that you made is just so clear, and you're right. There really isn't a lot of research about, especially the environmental, issues that are related to these public health things. So, my goodness, how does it feel to be a real leader, in a field that doesn't really exist right now?
It's growing. I mean, I've interacted with a number of, for example, politicians. Keith Ellison, who's a U.S. congressperson from Minnesota, who's been a real leader. Henry Waxman's been a real leader, some of our own representatives in California. And I think part of the problem is exactly what you just described. If you're not experiencing something, you don't necessarily think about it. So it's not active racism, it's that you're not thinking about, oh, we're going to put this plant in a poor community, which just happens to be 80% African American. And in the scientific level, because you don't have many people working in the area, they're not thinking about these they're not specialized problems anymore.
Right. So how do you make the argument to folks, in the scientific field and outside of it, that this kind of rigorous study is incredibly important, not just for the communities that are being affected, but for the entire country and world?
Yeah, they're micro. It's the same thing when people say, well, who cares about frogs? I say, well, look, if these pesticides only affected frogs, look at their integral place in the ecosystem. So let's say you don't care about frogs, but the frogs are eating the mosquitoes that carry the malaria. And so while you're spraying pesticides to kill the mosquitoes, you're also killing the frogs. The mosquitoes are becoming resistant, but now the frogs are gone as well. And then I say, well, look, if it's happening to frogs, it's happening to fish. What you care about commercially, what you care about because you eat. If it's happening to fish, it's in the drinking water. And so you work your way up. And you want to start, I think, with the more sensitive organisms, because if you protect the most sensitive organisms, then you're going to protect everybody. And so you can make the same argument, even if you don't care about, and I don't mean don't care, but if you're not thinking about, the higher mortality from breast cancer in Black and Hispanics, if you protect the most sensitive people, then by definition, you're going to be protected as well. So those are the ways that you have to think about it, I think.
What's next for you?
We're continuing to work in Africa. We're, the science has expanded. I'm very much involved in, there's a bill in Congress right now to ban atrazine, to regulate chemicals differently. Some of you may have heard about bisphenol A and plastics. And so just really trying to raise awareness and get people and get other scientists to become politically involved in disseminating and making the information available to the public and to politicians and to legislators, and not just sort of living in the ivory tower and not reaching out to really do what I think is what we were really designed to do, and that's to disseminate information and not lock ourselves in ivory towers with little specialized publications that you can't get unless you're part of that.
And I wonder who reads them, but that's a whole other story.
We trade them with each other.
Exactly. So your grandma is so smiling right now. So thank you
Thank you
very much.